and welcome to the Marine Biological Association's podcast, The Marine Biologist. This podcast is the accompaniment to our magazine, also called The Marine Biologist, and it explores the current issues that face our ocean. If you'd like to find out more about the Marine Biological Association, or where you can get your hands on the Marine Biologist magazine, then please visit our website, www.mba.ac.uk. Enjoy the episode. Hello and welcome to this episode of the Marine Biologist podcast. My name is Kathy. For those of you that joined us before, you'll have met me before. If you haven't joined us, please catch up on some of our other episodes. I'm going to pass you over in a moment to our other host, Guy Baker, the editor of the Marine Biologist magazine, who's going to introduce today's guest.
Okay, thank you, Kathy. Today we're going to be talking to Kes Scott-Somm, who wrote one of the articles in the most recent edition of the Marine Biologist magazine. And the article was about the Darwin Tree of Life. But before we go on to that, I'd just like to introduce Kes. Kes is a research assistant on the Darwin Tree of Life project, and she works on collecting, identifying and processing marine organisms. She's had a varied career working in environmental science and conservation. She loves science communication, and she's picked up all sorts of skills along the way, from electrofishing to diving. And she loves, as I said, communicating science in the natural world, preferably looking down a microscope. So welcome, Kes. It's great to have you here. Very excited. It's a fascinating project. And I guess, first of all, please, can you introduce the Darwin Tree of Life project?
Hi, Guy. Yeah, absolutely. So Darwin Tree of Life is all about sequencing genomes. For those of you who haven't heard of this before, we can talk a little bit more about genomes later on. But it's a very ambitious project. It's kind of a pilot project for larger and more coordinated efforts, looking at how we can try and start to sequence some of the 70,000 different organisms that we have in the British and Irish archipelago. So it's a really cool, exciting project that I've been really excited to be a part of.
Thank you. That's brilliant. And we were having a little, Kathy and I were having a little conversation before we started about the scope of the project, and that you're sampling eukaryotes. And we were having a very basic biology discussion about what that meant. And I was explaining in learned terms to Kathy what that meant. But I think it might be best coming from you, because you sort of enlighten us on what the scope of the project is and what is and isn't in it.
Yeah. So one way that we try and distinguish life on Earth, this is like one of the many ways that life is classified, is into these two different cell types, so eukaryote or prokaryote. And the majority of life is actually a prokaryote cell, so that includes things like bacteria. And if you look at any large tree of life, which is how we classify all life, most of the life on there is a prokaryote cell. Eukaryotes have a nucleated cell, so they're a little bit different, and that includes everything, including us. So that's all animals, plants, fungi, and also a group that we refer to as protists, which is kind of everything that has a nucleated cell that doesn't neatly fall into one of those other categories. So it's actually a smaller portion of the world of life, the very rich world of life that we have on this planet. But it's also a really interesting thing to look at. And prokaryotes are really quite difficult to sequence and to work with because they're so tiny. So there are other projects looking at that, but just for kind of practicalities and interests, we've got to start somewhere. We're starting with those higher branches, and then eventually people will be working backwards.
So the things that you're looking at on this project are the slightly bigger things?
Mostly, not necessarily. Some of the protists are single cells, and we're also looking at things like plankton, and some of those can be really tiny. But we're starting big and working backwards, basically. Like everything needs a different technique. And for the genome sequencing that we're doing, it's a really new kind of technology. It's a new kind of science. So the theory is we can get it down to working with single cells and getting a genome sequence from a single cell. But at the moment, we're still kind of in the research and the working process around that, making that happen, making that work. So yeah, one day, all the stuff, but for now, starting slightly bigger, I should say.
So it's the marine organisms of Britain and Ireland, so this sort of archipelago that we're in the Northeast Atlantic. And given it's so difficult to study marine life at all, why is it so important? And what might you find in the marine realm that you can't find on land?
Yeah, well, the project in general, Darwin Tree of Life, is looking at all terrestrial life as well, all life in that area. But at the Marine Biological Association and the laboratory here, we are very focused on the marine side of things. So we've been focusing on marine life, and it's important for a few different reasons. One is the variety. The marine environment is incredibly complicated. It's water rather than air. So you've got this much more 3D environment. And in marine environments, there's a lot more competition than there often is online because of that 3D-ness and that 3D nature. Things live on and around and inside each other in a much more complicated and ongoing way than sometimes they do on land. It's also, where a lot of the origins of life came from. So there's some really early evolutionary stuff that's happening there. From another point of view, in terms of sampling and looking at sampling it's very different. Marine organisms can be very different from each other. There's a lot of different body plans. So we need to approach it in a slightly different way to some of the terrestrial sampling. And it's also very unknown, the marine environment. It's an unexplored area, a lot of it still. And even in the UK here, we're still finding things out all the time in the marine world in a very different way to how we do on land. It's so much less accessible to us. So it does hold so many more secrets, which makes it really interesting.
So you're talking about collecting all these species and genoming and DNA. And can you just explain in kind of simple terms, what's the relationship between what are those things and why is it significant?
Yeah, absolutely. So DNA is kind of the recipe of life. So within your nucleated cell, you will have your DNA all tightly curled up on top of each other. And that holds all the information that makes you, and that makes all the other animals them, and makes up all of the things that they make. So it's the ingredients to start your life, and it's the ingredients to continue your life. And the genome sequence is all of that information in a really detailed way. So we can look at DNA in different ways. In part of science, we do something called barcoding, which is like a little snapshot of the DNA, which allows us to stamp what species that animal is or whatever. But with the genome sequence, it's all of the DNA information, all of the information within that cell, all of the whole sequence of it. And with that information, we can find out everything about that animal or plant or fungi, how it exists in the world and how it functions within its ecosystem. So it's really, really massive and high quality data set. About a few years back, the Human Genome Project sequenced the human genome for the first time. And that was a massive, massive push forward in science. It took many billions of pounds and many, many, many researchers and a lot of years. And the human genome is about 3.2 billion bases of DNA. But there are other organisms we have that we're collecting for the project that are much larger than that. Mistletoe is actually the largest genome sequence in the UK. It's more than 40 times the size of the human genome. So there's all sorts of strange and wonderful things going on, and having all those genome sequences gives us this huge amount of information that we wouldn't have had otherwise. So yeah, that's hopefully explains the DNA genome sequence bit.
That's amazing. I can't believe that mistletoe is so much bigger. The recipe for making mistletoe would be so much bigger than the recipe for making a human. There's no kind of consistency in organism size or complexity that we've kind of been able to work out. It's a bit of a mystery.
And what baffles me is, how do you get from molecules up to what the role of an organism is in? I mean, can you do that? Can you learn from the genes of an organism what its role is in an ecosystem, and how likely it is to go extinct? And these bigger questions, the things that maybe people can relate to a bit more, like pollution and climate change and things like that. How do you get from molecules to that?
Yeah, well, it's kind of like you have the ingredients book or the recipe book or the instructional booklet for that organism. So the hope is that we can start to see patterns once we have all of that information. Some of those questions are massive, and we don't know how the genome sequences are going to affect or change those. We have suspicions or hopes or some information from preliminary research, which means that we might be able to find the answers to some of those questions with the genome sequences. But that's part of getting that information in place. It's a bit like creating a library, and then all of the scientists out there can go and pick it up and find out. So we're hoping that we can maybe find out about things like potentially there are certain genes that are more prominent in invasive species, and maybe then we can predict invasiveness in certain species and change the way that we manage species by using that information. We can start to look at the difference between individuals, like what organisms are doing better in the face of climate change or worse, and is there anything that they have in common in terms of maybe certain genes that they have that we can start looking at. So the possibilities of the practicalities on understanding our environment and our ecosystems is kind of endless, and it is a bit baffling. And I think as one person or one individual researcher, it's quite hard to grasp hold of. It's going to be a mass effort. Collecting this information is the first stage. Understanding, decoding, and using it is the second stage, and that is going to take a whole army of scientists to be able to do that. So it's an ongoing thing.
So you've kind of alluded there to the fact that this is not just you doing this project. What is the scale of this? How many scientists are working on it?
Yeah, it's big. At the moment, it's around 300 to 500 scientists, depending on what's going on. We're about 10 to 11 different organisations working on the Darwin Tree of Life project across the UK. We're also we kind of partnered and collaborating with other projects like the Earth Biogenome Project and other big projects that are looking at genome sequencing on even bigger scales. And yeah, it's a lot of communication and collaboration. And apart from just the research organisations and the scientists who are directly involved, we have volunteers and other people who are also involved in volunteer organisations like CSearch, who collect some material for us and are very experts in their areas that we collaborate with. We've just had the University of Bergen, people from there here coming to help us to identify hydroids and work on hydroids. So it's a huge, huge network of different scientists, and it would be hard to put a number, I think, on all of the people that the DToL project has touched or is connected with. And just here at the MBA, we have researchers that aren't directly a part of Darwin Tree of Life, but are connected in that they're using the data already as part of their research. So it's already being utilised by scientists in a very far-reaching way.
Mm. And is it very interdisciplinary, or is it chiefly biology?
I would say it's incredibly interdisciplinary, because there's this whole aspect of things like data management, utilising new areas of technology. There's the whole communication side and public engagement and outreach and all these things that are connected to just the raw science. There's even just within the work that we're doing within our lab here, it's a collaboration between molecular scientists and ecologists, and all of these different people coming together to collect and understand because when you start going out into the environment to collect animals for genome sequences, you need people at all those different stages and all those different places. And it's also just been about new ways of collaborating and working with each other across different organizations, because it's very unusual in science to have these kind of projects that are huge collaborations across multiple organizations of people working really, really collaboratively on the same thing. It's often organizations will each have their separate role that they're doing, but we do a lot of stuff very collaboratively. So there's lots of ways that it's interdisciplinary, and that it's involving different types of areas of work.
I think one of the things you said there is about the fact that you've got, molecular biologists working alongside ecologists, and that's really not very common, is it? Within this industry. Has that caused any challenges, any particular, difficulties that have come up that you've had to overcome?
Yeah, I mean, absolutely. From my point of view, I'm much more of an ecologist than a molecular person. And there's a very different level of cleanliness between the two, because there's ways that you can contaminate DNA without meaning to, because so much of it is so very small, you can't see what you're doing. So molecular scientists approach things in a much more meticulous and clean kind of way than some of us ecologists are used to doing. So yeah, it's been a real learning process for me of what I can touch with and without gloves, and what things, and some things have need to be replaced or moved, or we need to change the way that we do things to ensure that we're not having contamination happening. So yeah, it's definitely a learning process, and we're just so lucky that we've got such good colleagues here who can kind of talk us through those things. And for them, it's a bit like explaining to a toddler what's happening, but they've been very, very kind and calm in helping us get it so that we're working in a more molecular way, in a way that's more cohesive with what they need.
Oh, and I'm just thinking of the title of the article, Darwin's Tree of Life, the title of the project. I was just sort of thinking it's interesting to sort of look at this subject of evolution and how species are interrelated, and how Darwin first pieced together these interrelationships through looking at finches on Galapagos Islands and things like that. And I mean, what's it like to sort of reflect back on what he was doing, almost working blind and coming across this field that still holds so much sort of fundamental truth, it seems, even when it's being pulled apart to the molecular level.
Yeah, I mean, that tree of life concept, which Darwin really popularized and really kind of brought life to, pardon the pun. But yeah, I think in a way, we do have all this new information, new work that we're going on. And in another way, we're still kind of are stumbling around in the dark with science. You get to a new level and there's just so much to know, and so much that we don't know, and so many unknowns, that I still feel like we're at the very beginning of science. But I do think it's kind of funny that it's called the Darwin Tree of Life project, because really what we're doing is continuing the work of Linnaeus, who first categorized everything and gave those binomial names, the scientific names that we use, and started putting everything in their place. And the Linnaean project was that first project that started to name everything in nature. And we're kind of continuing that by then putting the genome sequence behind those names. And Darwin was about putting those in their place on the tree, which is almost something that's almost a step ahead what we're doing right now. It's like we'll then be getting to grips once we have the data of where everything fits and how everything fits together. So we're almost still a step behind Darwin in some ways, it feels like, to me anyway. there's so much still to be learned, and yeah, his way of looking at things is still very much how we're doing things, just working in a method, and trying to make things work, and seeing what results we might have coming out the other end. So yeah, science is much the same as it's ever been.
So does that mean some of the work that you're doing might mean that some of those things move around on the tree?
Yeah, absolutely. I mean, the things moving around on that tree all the time. It's a constant changing tree, and almost it's a web, because things can go backwards, or things evolve in different ways and de-evolve, and things are changing and genes are going left, right, and centre. So a web or a net is kind of a better way of thinking of it sometimes. But yeah, absolutely. Things change on that tree with or without our help, but we will definitely be making things change around on that tree for sure. Yeah.
And they change just because we simply learn a little bit more?
Yeah, we learn more about something, or there's a concept within taxonomy, which is the area of naming things, where we will suddenly find a cryptic species, so two animals that maybe look, or plants or algae or whatever else, that look very similar to each other but are actually genetically completely different. Good examples of things like that are things like the hedgehog and the tenrec, animals that look identical but have evolved from very different ancestors in very different places. So we get new information on things. I mean, we still haven't discovered most of the life that's out there. There's still so many life forms still to be discovered, let alone named and labeled and put into the right places. So yeah, it's a very big part of science still, is just getting everything down where we think it might be.
And how do you discover a new species with these sort of techniques?
So we've actually probably found some new species already. We can't
oh, wow.
It's a long process to actually confirm that. But when you find a species that doesn't quite look like the right thing, or when you find a species that maybe has some different characteristics, or even that looks exactly similar, but the DNA and the genetics that are coming out are different, there are different ways to try and find out if something's a new species or not. And it's a much more involved and difficult process than I think even a lot of us working on the project realised. It's not, you find a new species, you prove it's a new species through its DNA, and then that's that. There's a longer process of naming things and working things out. So yeah, it's likely it's not the purpose of the project to find new species, but it's definitely something that is almost, if you go looking, you're going to find new species. So it's definitely going to happen, I think.
So how long will we have to wait until we have the Kes? Do you get to name them when you find them?
I don't know how that's going to work, because it's so collaborative. It's very rare that it's one person who's doing the collection, the ID and everything from start to finish. We're working so together. So I don't know how that's going to work. I don't think I'll get one named after me. Although we do come up with our own common names for all the species that we find that don't have one. That's so many species that we don't always do that. But if we think of a good one, so I have already named one of the starfish that we got through the project. I've given it the common name of the recursive starfish, because it's a starfish with a pattern of stars within it. So that's quite fun. We get to do some common names as well.
Nice, it's quite cool.
And how can the project directly benefit people? Other ways?
I mean, again, there's multiple ways, and it's complicated in that we're almost not doing the actual science ourselves. We're creating the resource for other scientists to use. So how that goes on to be used is dependent on the researchers that come at the same time as us and also after us. But the natural world is a huge amount of resource for humanity, and that's directly. So In the marine environment, we have lots of species that have been, their past genome sequences have been used to understand what new drugs we could use, and things like sponges and bryozoans, which are quite interesting animals. They've already been used to synthesize new drugs for things like antibiotics, anti-cancer medication. So there's a huge potential for new medications, specifically antibiotics, which is a big problem area for the future of medicine. We have so much biotic resistance happening and so many changes to that. So having new antibiotics options is going to be a really big part, I think, of some of the outcomes or potential outcomes of this project. So there's kind of the direct science, and it's not just medicine. For example, there's a marine worm that we have called a quill worm that makes a case for itself out of a very see-through, hard material that looks very like plastic and is not plastic and biodegrades very quickly. But if we could understand how that worm makes that, we could make a new type of plastic product that's not polluting in the same way. So there's all these potential benefits to technology and the way that we live our lives. And it's even more complicated than that, because even the way that we've set up new data and created new data portals and new data platforms could have an impact on the way that we manage data in other areas. Things like the NHS here in the UK could, benefit from the way that we're managing some of the massive data that we have here, because one single genome sequence holds so much data, and that's all being put up online and managed in a certain way. So even the way that we're doing those parts of the project could have a benefit for society, for humanity. So it's kind of a never-ending. It's what other people make of the project almost, rather than what we're creating in itself. There's so many potential benefits.
It sounds like an overwhelmingly large amount of possibilities.
Yeah.
Amazing. Yeah.
That's definitely how I think it feels to all of us. I mean, if you watch any of the Darwin Tree of Life stuff from anybody, I think all of the scientists working on it definitely feel like that. Just it's like a fundamental part of science to be doing this work, and what it will create is beyond our ability to currently understand.
And that's exciting.
Very, very cool. Yeah.
So how long, I think you talked earlier about it being, is it 70,000 species? How far through that process are you? How long is that likely to take? How much of it have we done?
Well, when you go on the website, you can see, because that's a really difficult question to answer for a few reasons. And one of them is that it isn't a one-stage process. So when we collect things, that's the first part ticked off. Then when we fully identify them and make sure that we know what they definitely are, that's the next part of the process. And then when they get sent off to have their genome sequence, that's the next part of the process. But then once they've had their genome sequence, that genome needs to be curated and put online, and a genome note needs doing before they're published. So it's not, there's like different targets at different levels for the numbers, and those numbers change every single day. We have new genome notes going up on the website every day. We have new species we collect every single day. So those numbers are constantly changing. From the kind of other side of it, the longer this project goes on, the harder it will get to finish, because the more rare species are going to become. So we're going to be going after tiny little marine worms that have only been seen in the sea in the UK, but not a long time. Or we're going to start going after things that are much, much rarer in the UK. So the first stage of the project has been about collecting those larger organisms and those easier to find organisms. And then as we go on, we're going to be going smaller and smaller and rarer, which is going to slow the project up. It's much more difficult to dissect out a tiny little hydroid, as we were doing last week, than it is to dissect a big fish or something. So different things take different timescales. So it's really difficult to put precise numbers on. I think the best thing to do to answer that question is to go to the Darwin Tree of Life website and see how many genomes we have up at any one point. And yeah, we definitely have a long way to go, and we have a huge backlog. We're over 700 species from the laboratory here and from all the marine stuff. So, and then the main focus for the beginning of the projects was insects. So there's a huge amount that have their genome sequenced, and Lepidoptera in particular, there's a lot of genome sequences already published, but it's just an ever-changing number, I think. But we'll get there, or at least get far enough through it that it will be really meaningful. I think already the research and the amount of data that's there is meaningful for scientists who will need to use it. There's no end.
And do you get thinking about the deep sea, and there's some areas of deep sea in UK and Ireland territorial waters, and I mean, are the research groups going out dredging or doing sampling anyway, and the Tree of Life project sort of connects with those to get samples brought back? And also, if there's, sorry, two-part question, say someone catches a rare shark or something in the deep sea, is there a way of the Darwin Tree of Life project going, oh, can we have a little bit of that to stick it into our database?
Yeah, kind of the answer to both of those questions is the same, because it depends on what collaborations we have. So our colleagues at the Natural History Museum have recently gone out on a deep sea voyage, and they were just lucky enough to be allowed to tag along to that research. So while on board, whatever came up, they were able to take a sample from if they needed to. But again, it's almost about like maximizing what we can do with the resources we have. So like, who can we collaborate with? How can we get that? And there's priorities, you know, as there is with any project. We're prioritizing either areas where we already have connections, or areas where we have strong research interests of the scientists already working on the project. So certain things are being prioritized for those reasons. But yeah, whenever we can get connections. Deep sea surveys are really expensive. Getting out into the deep sea is really expensive, and it would be a massive drain on our resources, which might mean that we can't put so much into catching butterflies or something, which would be, you know, a shame, because in the eyes of the project, those things are kind of equal. There's equal interest in the different things. So yeah, it would be cool to do more collaboration with things like that, and to get to these more difficult areas. But for now, there's so much we haven't caught in the shallow seas that we're still focusing on that, and hopefully we can collaborate more in the future.
Can you tell us a little bit about, you know, how did you end up doing this? How did you end up here?
I don't really know, to be honest. I've done a lot of different things. I think one of the things with the Darwin Tree of Life project, and particularly with the job that I'm in now as a research assistant, is it's so varied. You know, I have to do everything from going out and doing field work, identifying stuff in the lab, dissection, like edging onto those molecular things where I need to know how that works so that I don't mess it up. And then this side of stuff, like a huge part of this project for me has been about communicating this project and understanding this project, and trying to push this project, not even just to the general public, but to other scientists, other researchers, try and understand how important this project is. So I guess the reason that I particularly have ended up in this role is because I had that varied background. I worked in consultancy and did a lot of taxonomy and ID. I've worked in comms and done a lot of comms stuff and connecting with people and stuff. I've done a lot of field work as part of previous jobs and as part of my degrees. And so having that kind of wider basis of knowledge to be able to connect those things together, and working with a huge data set. My past project was on one of the biggest citizen science data sets that there is, and dealing with all that data and managing it and communicating it. So having those different skill sets has been a real boon to me. And that's often not the case in science. Often the case in science is the more specific you are, the more desirable you are. So I've always been moving around and not quite settling in one place. And for the Darwin Tree of Life project, that's actually been a good thing, having that.
Collaborations by their nature are varied and require all sorts of different skill sets. That's really positive. And where next in your career, Kez? What do you see yourself doing down the line?
I think one of the great things about this project is it has got me, has allowed me to try out some different areas and some different things. And it's kind of made me rediscover my love of taxonomy. Taxonomy is a weird one because it is seen as this older science. It's the science of Darwin and the science of Linnaeus and the science of these scientists that went before us. So there's not as much cutting edge excitement around identifying something. But it's such a skill, and it's such an interesting skill to develop. And it's been the part of the project that I've enjoyed the most, getting that real variety on looking at so many different organisms in so many different shapes and sizes and body types, to try and identify them. But through realizing that was my favorite part of the project, I'm now going to move into doing that as my permanent job. In the plankton team here at the laboratory, we've got the continuous plankton recorder, which is the longest running plankton analysis. And so I'll be going into that and doing my plankton identification. So getting more into the smaller stuff and more into the detail and more into the microscopes than ever before. So for me, it's been really good in guiding me more into that role. I always knew I enjoyed the comm side. I always knew I enjoyed the communicating work side. I always knew, and field work can be really fun or really terrible depending on what the weather is like and how long the day is. But being under a microscope, like having a look at all the tiny little things and seeing them on that different scale, where you suddenly appreciate something that just looks like a little ball of snot or something, and then you look at it up close under the microscope and it looks like the most amazing thing you've ever seen. Working on that detail is something that's what I'm looking forward to for the future.
Mm-hmm. Sounds exciting. Sounds like you've had a fairly varied career up until this point, and to just continue to find the bits that interest you and to keep making that journey in a different direction is really cool. So as someone that's done that, then, if there was anyone listening to this who maybe is in an area of marine biology and would like to move, or maybe isn't yet working as a marine biologist but would like to, or is interested, do you have any tips, any advice for somebody in one of those situations?
Yeah, I think one of the things that probably most people get caught up in when they think about careers is things like what job is right for you, or what title is right for you, or what direction is right for you. And they think so much less about what skills do I want to learn, and what day-to-day things do I enjoy doing, and what day-to-day things can I do continually every single day. For me, I realised relatively early on in my career that doing 9 to 5 looking at a computer just didn't suit me. And I also realised that field work, being out all the time in all environments, also doesn't suit me. I find that too draining and tiring. Whereas lab work and sitting under the bright lights in the lab looking at stuff, that is something that I don't seem to get bored of, or don't. Everyone has a day where you think, I don't enjoy this right now, or I'm not having a great time, or I'm bored, or I'm frustrated, or this isn't working. But thinking more about like what actual things do you physically like doing was really important for me to help me figure out where I want to be. And also what skills do I want to learn? Because the world is changing all the time, and the things that get funding or don't get funding, or where the money is changing all the time. And although none of us get into marine biology for the money, there is an element of, you need to factor that in. What kind of lifestyle do you want? How do you want to live? Do you want to be able to pay all your bills? And, you need to consider that. So what skills do you have that maybe are transferable if things do get tough? Or what things can you take into different careers? Things like molecular science, they're so applicable across so many different fields once you've got that experience. So if you enjoy that meticulousness and being almost following recipes and being really precise with things, and having a relatively good level of maths, those things are all important for that career. So it's kind of understanding not just what you're good at, because what you're good at can change. You can practice and you can learn. What's more important, I think, is what you can do all the time without getting bored, or without finding that you're losing your interest. Like, do you need to always be learning? Because for me, I do. I need to always be learning something new. Taxonomy is an area where you're always learning, because you're always finding new animals to look at, new creatures to look at. Or, like I say, the taxonomy's changed, so you need to learn it again. Things change name or change group, or so for me, that's what's important, and everybody's different. So thinking about those things rather than what job suits me. But definitely not forgetting the money side, because I think we don't talk about that a lot. We always say, go for your passions, go for what you love. But you do need to think about what life you want to lead. Do you want to have holidays and go traveling? Or are you really content where you live? And those kind of things are all important to factor in.
Well, thank you so much, because I'm sure we could ask you so many more questions about the project and about your career and about, where you're going to and where you've been. But that's probably all we've got time for today. But if anybody wants to carry on the conversation, they want to get in touch with you, they want to follow your work, where can they do that? How do they do that?
I use Twitter and LinkedIn, like all good researchers. I also have a ResearchGate profile. I can share all the links to all of that stuff in the podcast notes if anybody's interested.
Excellent. Are you happy to share your Twitter handle with us now?
Yeah, absolutely. It's Casella Nutella, which probably needs spelling out for you, but I can write it down later as well.
Excellent. Well, thank you so much for your time and for sharing with us about the project and what's going on. And maybe we should hear from you again soon.
Absolutely. Thanks very much.
Thank you, Casella. That's brilliant. Lovely.
Yeah, thank you for having me.
Thank you very much for listening. This has been the Marine Biologist podcast. If you've enjoyed this episode, then please check out our other episodes. Please subscribe to our podcast. Please rate it, share it with your friends and family, and let us know what you think. And we will look forward to welcoming you to the next episode soon. Take care.